see, this is the title of my talk, Exocellular Vesicle-mediated mRNA Therapeutics. And the next slide shows the, could I have the next slide? Yes. Yeah, shows, a lot of people have collaborated, many of my Stanford colleagues, elsewhere as well, and their names are here, and I don't have the time to mention. People at NIH contributed as well, and you can see their name, but I won't describe them. So we, use this therapeutic approach initially to treat cancer by prodrug therapeutic approach. As you might know, prodrugs are inactive in their native state, but they become highly toxic upon reduction catalyzed by a bacterial or viral enzyme, and they kill cells by generating DNA adducts. And this process is called gene-directed therapeutics, gene-directed, GDEPT. Could I have the next slide, please? Good, no, go back, that was the missing part, yeah. The important thing about prodrugs is that they target both growing and non-growing cells, and which is an advantage because many tumors have non-growing cells. And the examples are CB1954, also called Tretazicar, and the prodrug and its activating enzyme that we ourselves discovered, called CNOB and CHRR, the enzyme. And as I said, it's called gene-directed prodrug chemotherapy. Next slide, please. Okay, two prodrugs have been subjected to clinical trials. One is the genciclovir TKH1 enzyme. It was subjected to a phase 3 clinical trial. And the other one, CB1954 or Tretazicar, phase 1/2. But the results were extremely disappointing. And if I could have the next slide, it could indicate why the, it was not effective. One reason was insufficient gene delivery and expression duration. The enzymes, the wild-type enzymes used were not potent enough. Viral vectors that were used were problematic because of immunogenicity and potential pathogenicity. And there was no selective targeting. The gene for the encoding the enzyme had to be injected directly into the tumor, and this limits applicability because, as you know, not all cancers are accessible and can be, can be used for direct injection. Now we have addressed these issues, and this has been greatly helped by our discovery of this prodrug that I just mentioned, CNOB. The one important thing about CNOB activation is that the drug it generates, MCHB, is highly fluorescent and can be visualized. So that really enables you to make sure that the drug generation is confined to where you want it to be confined. Next slide, please. And this is the detail of, so this is the prodrug CNOB, and this is the enzyme HCHRR6, and this is the drug. And then, this gives you details of fluorescence and things like that. And then we worked out the mode of action by looking at binding of the DNA to mitochondria and things like that. And if I go to the next slide, it shows you the fluorescence. You can visualize it in monolayers as well as spheroids, as well as inside the tumors. As you can see, it is confined to the tumor. And we also have characterized as PK/PD. And the next slide gives you the meat of these experiments. MCHB, the drug that is generated from the activation of CNOB, causes mitochondria-mediated apoptosis. As I said, it kills both growing and non-growing cells. MCHB fluorescence is quantitative down to 2 nanogram per gram tumor in living mice. So you can, as I said, find the localization. It has very favorable PK/PD, and MCHB, the drug, has an excellent bystander effect. What that means is that the MCHB can leak out of the cell where it is being generated, and so can kill neighboring cells, which is very important for the effectiveness, success of any gene mediated therapeutics, because it's not possible to transform all the cells in a tumor or whatever by the method of gene delivery. So this bystander effect, which this regimen has, for which this is very effective, is highly useful. Next slide, please. Okay, so I mentioned that one of the problems was that the enzymes were not potent enough. So the enzyme that we discovered, which activates CNOB, initially it was called CHRR, but we have now humanized it and modified it by using all sorts of procedures, shuffling, error-prone PCR, and crystallization. And in the end, we found that the enlargement of the active site of the enzyme, which contains FMN, and it's a tetramer, is strengthening the dimer-dimer interaction by making suitable changes at the interface of the dimers. It is the key to improving the enzyme, and our enzyme is 100-fold more active than current nitroreductases that are being used to activate prodrugs. Next slide, please. Okay, so the next thing is improved gene delivery. And as I think most of you are convinced, even though DNA has been extensively used for gene delivery, the mRNA is superior for this purpose. For one thing, DNA must enter the nucleus for transcription before generating the desired protein. This process is highly inefficient. A very small amount of cytosolic DNA enters the nucleus, and then DNA poses the risk of insertional mutagenesis. And as I mentioned, mRNA, in contrast, is directly translated upon cytosol entry, and it poses no risk of insertional mutagenesis. It has stability and efficient translation issues, but there are many methods available to counter it. UTR and the coding sequences can be modified. Circular RNA can be used, and base substitution like pseudouridine and such, and capping and all that can be done to stabilize the mRNA. Next slide, please. So, people have been very successful in using lipid nanoparticles. And as you know, the advantage of LNPs is now greatly highlighted by these COVID-19 vaccines. And I have something more to say. The LNP, lipid nanoparticles, form a kind of a structure, tertiary structure, that protects the mRNA, and it has, and they have many other advantages. It can be easily targeted and so on. But our own emphasis has been on exosomes, or extracellular nanoparticles. These are non-immunogenic for the most part because they are nature's own antigen delivery system. Exosomes are used to transfer information between cells at a distance. The tetraspin CD9 fuses with the cell and directly delivers its cargo into the cytosol. They are small, avoid clearance by the RE system, avoid liver, lung entrapment and resulting toxicity, and the endosomal pathway and so on. And also it has been shown that HEC293 exosomes are completely harmless in mice. But the problem is that nobody really had succeeded. You know, exosomes have their own mRNAs, small siRNAs and proteins and all that. But attempts to introduce a foreign mRNA had consistently been unsuccessful, even in our own hands. But finally, we succeeded. Could I have the next slide, please? Let me first show you what the EVs look like that we made. We made the EVs by differential centrifugation, and you can see what they look like by nanosite sizing. And they range between 30 to 100 nanometers, and the same size is indicated by transmission electron microscopy. And you know, there are accepted standards as to what constitutes EVs or exosomes. They should have at least 3 proteins, and here you see CD63, CD81, and MFG-E8. So they are genuine exosomes. Next slide, please. All right, so having made these exosomes, the next issue, there are 2. One is to target them to the cancer, and for our proof of concept purpose, we have used treatment of HER2 positive breast cancer, which, as you know, overexpresses the HER2 receptor. And so what we did was to make a special protein, a chimeric protein, which is shown in here, which contains the leader sequence to permit the protein to get out of the, to get to the surface of the exosomes, and then joined to an ML39 SCFV, which has a very high affinity for the HER2 receptor, and then this lactatorin C1C2 domain, which binds to phosphatidylserine, which is expressed on the surface of the exosomes. And this shows you the structure, the bending of the protein, which we call EVHP, and this is its predicted tertiary structure. Next slide, please. Next slide. You can see that my postdoc. Okay, so the next thing, as I said, is how to put the mRNA, and the mRNA we wanted to put into the exosomes was the improved was the mRNA that encoded the improved HCHRR6 mRNA. And for that, in collaboration with certain other people, we made a special plasmid which contained the zip code. It's a nucleic acid sequence that is supposed to promote entry of mRNA into the exosomes. And in fact, we did succeed using this approach to put the mRNA into the exosomes. And these exosomes were able to deliver the mRNA in an effective form, which I'll show you in a minute, into the recipient cells. But having a plasmid, first of all, you have not got rid of DNA, because plasmid would have to get into the nucleus as well. And secondly, it can be harmful to the patients. So now what we do is to use IVT, the in vitro transcribed HCHRR6 mRNA. And, it proved quite effective, as I will show you. And also, even though we did the initial characterization with CNOB because of the visualizability of its drug to make sure that the approaches we were using were successful in confining the generation of the drug into the tumor. We have now shifted to CB1954, also called Tretazikar, for the reason that it has already been subjected to clinical trial, which I had showed you a little bit earlier. And so as a result, a safe human dose has been established, facilitating translation to the clinic. Next slide, please. This is these are the steps which we used in making EVs containing the HCHRR6 mRNA, IVT mRNA, and then this is the exosome containing the mRNA. Then we mix it with this purified protein, EVHB, and because of phosphatidylserine, they they're decorated, they're exhibited on the surface of the exosome, and so they can then attack. They can then combine with cells expressing the HER2 receptor, inject the mRNA where the enzyme is made, and then if you have CB1954 or CNOB, then there is cell death. Let me make one more point. This enzyme, HCHRR6, is effective not only in activating CNOB, but also CB1954, which proved to be a great boon, because as I just stated, CB1954 has been in clinical trials, and we know it's a safe dose. Next slide, please. Okay, so as you know, naked mRNA is subjected to fragmentation. And even though we took many kinds of measures to try to stabilize it, still, throughout this highly extensive scheme for making the EVs able to deliver mRNA to the cells, I showed you the various steps. It was necessary to make sure that at each step, the mRNA remained competent to be able to activate CB1954 or Tretazikar. Now, to do this with CB1954 itself was impossible almost, because its drug, to assay it requires very complicated procedures. But what we did then was because the enzyme can activate both CNOB as well as CB1954, all we did at every step was to make sure that the protein encoded by this mRNA was able to activate CNOB, which we could very easily visualize. So vicariously we established, and throughout all these stages, that the mRNA remained competent to activate CB1954. Next slide, please. Next slide. Yeah. Okay, so, it turned out that the EV made from the IVT mRNA actually contained more mRNA than the plasmid ones. And whereas for the plasmid ones, we needed 5,000 EVs to deliver one mRNA copy, with the IVT, we needed only 50. Next slide. And this shows you that the IVT mRNA was inside the exosome and not just hooked on or adhered to the surface of the exosome. Next slide, please. So this is the in vitro characterization of this whole procedure, and it established that functional mRNA was delivered specifically to HER2 cells, and we did it by ELISA, by flow cytometry, by determining functionality in the presence of actinomycin D, and since the effect was not inhibited by actinomycin D, we ensured that it was mRNA that was the effective agent and not DNA, because actinomycin D inhibits transcription. Could I have the next slide, please? And then we also wanted to know what the efficiency was. So you see, now we have these IVT mRNAs. We've got these exosomes containing IVT mRNA, and exhibiting the HER2 targeting protein. And what we did was to determine how many copies of mRNA were contained in the EVs that we were using, and how many were received by the cells. And we found an efficiency of 50% at a very short time. And the reason, I believe, is that the targeting efficiency is very high. Next slide, please. So this summarizes the points I've been making. Actinomycin D does not affect expression. Therefore, these exosomes deliver AChR6 mRNA and not DNA. mRNA is inside the exosomes, can generate its translated product, can generate MCHB fluorescence from CNOB, which shows that it is also capable of activating TRETA-Z-CAR. Okay, and the transfer efficiency from the exosomes into the recipient cell was over 50% at a very short time. At a longer time, it was even higher. Next slide, please. And now here is the crux of the matter. This is in vivo test. So what we have here are xenografts of HER2 positive breast cancer orthotopically implanted in mice. And we treated them, with several controls. All these are controls, but this is the curve, the blue one, that shows that the growth of the tumor was completely inhibited after about 8 days or so. And the point is that even though the last dose was administered on day 7, for the rest of the experiment, which lasted for 34 days, there was no growth of the tumor, which clearly indicates that the tumor was killed. And there is the Epr effectiveness, which we had just discussed, that's conveyed by this yellow curve. You can see that there is some effectiveness just by, these are non-directed EVs, but they can extravasate because of EPR and are partially effective. Next slide, please. Okay, so what we have found so far, and what I have showed you so far, is that we call these exosomes that are directed, that are showing the HER2 targeting ligand or protein on their surface, and contain the AChR6 mRNA. These are called exodepts. And these exodepts have clearly succeeded in killing the implanted orthotopic tumor in HER2 positive tumor. But that does not prove that no other side effects occurred, because ERBB2 is expressed by other cells, and that can be recognized by anti-HER2 targeting ligands, and so it was possible that the mRNA was delivered to normal tissues as well, and that there was a general toxicity. So to explore this, We did extensive hematology and serum chemistry analysis, analyzed these parameters, and the details are given in here. Lots of the parameters were examined, and no toxicity was found in any of this case. Very minor elevation of liver enzymes, but that happens with CB1954 alone, and it is very transient. And next slide, please.
Professor Mateen, also about 2 more minutes. Okay.
Yes, I shall be finished.
Great.
And these are the kidneys and all that, and again, no effect at all. Next slide. This just shows you the data, which I have already summarized. Go ahead, please. Next slide. Once again, the same data. Okay, the next step is to, all of this was in immune compromised mice. And now we are looking at immune competent mice. So there are mice that spontaneously develop HER2 positive tumors in an immune competent situation. And we think, although we don't know, that the immune response to the human elements or mRNA or whatever might actually make this therapeutic approach even more effective, because as I just showed you, no side effect occurred, showing that the exodepts were localized entirely to the tumor. And so any immune response that is arisen is likely to be directed to the tumor, enhancing the effect of this therapy. Next slide, please. And that I think is, okay, EVs can cross the blood-brain barrier, and therefore they can be used for this purpose as well. And I think, next slide, please. Okay, as I mentioned to you, we used HER2 positive cancer for proof of concept, but this technology can be used to treat any cancer or disease in which a ligand or a receptor is overexpressed. All you need is a proper SCFV or targeting moiety, which can be attached to the exosomes by precisely the same methodology that we use for this EVHB protein synthesis. Next slide. Okay, so this is the lipid nanoparticles, and I don't think I need to go in there. I think that these have really, previous slide, these have a great future. As you know, they've been very successful for the anti-COVID vaccines, and they can protect mRNA, and they can be easily targeted, by having pegylation and things like that. So that's the end. Thank you.